be what is allowing that virus to attach to its host cell. You also see here M protein, which is the orange, all right? And then you have this yellow E protein, all right? So as best we can tell, the M protein is helping direct viron assembly, right, or the assembly of an individual viral particle. And we'll talk about viral assembly here in just a little bit, but if you think about it, a virus invades a cell, it makes all these different parts, and then you have to have a way to kind of put them together, right? So this M protein helps put them together. And you'll see here in just a second, the E protein is really interesting because it's going to essentially help the virus be able to bud away from the host cell. Okay, and I'll show you what that looks like here in just a second. Now, if you were to go and look up anything about vaccines, you would see that the majority of vaccines, right, are targeting this S protein. Now, they're doing it in different ways, right, but that's kind of the big point. If you can prevent any sort of attachment, then it doesn't matter what other capabilities or other proteins the virus has, right? You block the S protein, it can't attach. Okay? And again, I can't remember if what I have, I feel like I can't remember if I updated this on Moodle. If I didn't, I'll put this one up today so that you guys have this, the same lecture with this little bit of COVID extra stuff. The only thing that would be missing from the one you have on Moodle would be the COVID stuff. So, but I'll double check and add that today. So what's really interesting here and what this is showing you is that the virus is assembled in the endoplasmic reticulum and the Golgi apparatus. Okay. So again, the host cell, remember, viruses, right, can't survive on their own. So here, right, you see it using this host cell. And here, right, you can see, right, a new viral particle being assembled. So here's the spike proteins in the red, the green is the M proteins, right, and the orange color is the E proteins. And so what scientists are speculating, and again, there's still work to be done, is that the E protein does one or two things, right? And so there's speculation that it actually causes a fold or kind of a bend, right, which allows the budding shape to take place. And then, right, the idea would be potentially you have two E proteins that come together, and that kind of is what cuts off that newly formed viral particle and allows it to bud. Oh, and I'm sorry too, and I'll come right back to this. I forgot to. So this is an electron micrograph, or a scanning electron micrograph of a cell that's infected with the coronavirus. So these are kind of coronavirus, individual viruses right here, right? From this transverse section, they kind of look like little pizzas in there, right? And if anything too, sometimes you're like wondering, well, you know, what does a viral infection look like in a cell? So, right, if you got filled with something that you shouldn't be filled with, right, it makes normal cellular function kind of a big issue, all right? So any questions here? Do we feel okay about this? Again, I do want you to know ACE2 receptor, right? I want you to have, again, a very basic understanding of the M protein, S protein, and E protein. Yes, ma'am. It doesn't. Can you rephrase it a little bit for me? Oh, yeah. So remember, I kind of talked about this more, and we definitely see this a lot in enveloped viruses, right? So the idea of some of your structure as a virus is a lot of the host cell that you've infected. That makes sense? And so, you know, when you bud off, you're taking some of the cell membrane and other components kind of with it. So you're basically covering yourself in pieces of the cell that you've infected, right? So you kind of you're pinching off. Does that make sense? Okay. And what you'll see too is that, I can't remember if I have this on a diagram or not for you guys, but what's really cool is when a virus is replicating and it's getting ready to make copies of itself, what will happen is some of its proteins will be directed to the cell membrane, and it'll get embedded in the cell membrane, all right? And then that kind of serves as basically a target. So the rest of the virus is able to see, okay, the proteins have been put in the cell membrane here, so we go here, and then that's the part of the membrane that gets pinched off, all right? So that's a good question. All right, so now we're kind of doing, you know, a little bit of a about face back to kind of the original content. So some of the viruses that are easiest to study, and we'll talk about maybe briefly here in a second, have a lot of kind of maybe potentially future research potential, are bacteriophages, right? So now the good thing about bacteriophages is that they infect bacteria and not you at the moment, right? Again, things mutate and things change, and so I am certainly not going to go out on a limb and say bacteriophages will never infect you or infect humans, right? They possibly could. When we talk about bacteriophages, we have three different kind of varieties here. We have a lytic, a temperate, which is lytic and lysogenic. And again, I'll talk about what both of these terms mean here in just a second. And then you have, right, filamentous, right, shaped right here, okay? And again, don't worry about getting this all down because we're going to talk about each of these individually, right? So let's talk about a lytic bacteriophage, right? When you hear the word lytic, hopefully you're thinking lysis. Okay. And so this, right, infection is going to result in the host cell bursting open. All right? Again, think about it. You can only have so much room, right, in a cell. It bursts open, and then these viruses go and infect other, in this case, bacteria that are in the area. So a couple of things that I want you to note about these bacteriophages. In this case, we're talking about a lytic bacteriophage, right? It actually, right, well, first it has to attach, all right? And again, these kind of lunar lander legs, right? These are their spikes, right? That's what it allows it to find its target, right? Also note, right, that they, right, the caspid of the virus does not enter the cell. In this case, right, it simply injects its nucleic acid, all right? Now, along with that nucleic acid, it's also putting in enzymes, all right? And what happens here, right, is that the host chromosome, right, in this case, bacterial chromosome just has one, basically gets chopped up into little pieces. Yes, ma'am?
So if it's not the shape that looks like the man lander, is it going to be able to attach without those spikes?
Are you talking about like the filamentous one?
Like the helical and the iso.
Well, icosahedral is still going to have spikes on it, right? Right. If I mean, the coronavirus is an icosahedral, right? And it has those S proteins, right? It has the spikes on it. And the filamentous you'll see here in just a second. I think that'll maybe kind of answer that question a little bit, okay? Good questions, though. But yes, they're all going to have some type of spike. The spikes may look different, but that spike is what interacts with the receptor of the host, right? So the host chromosome gets broken up, which is good, right? If the host chromosome is broken up, then the bacteria cell is not spending as much of its energy on making copies of itself like it should, right? The virus can now utilize that cellular machinery, right? Those enzymes, those ribosomes, right? All that fun stuff to make its own individual components. All right? So they're synthesized here. So you're making more copies of nucleic acid. You're making more copies of the caspid, right? You then have all these individual components, right? And then they get assembled. And then, boom, right? That cell bursts and they go on to infect new cells, right? So the lytic cycle results in, I don't want to say immediate, but relatively immediate cell death.
So does that mean that it releases the viruses to other cells to do the same thing?
Yeah, so, you know, we haven't talked about it a ton, right? But I think you can appreciate probably from microbiology lab that you don't typically just have one bacteria that's hanging out by itself. Yeah. So what they're not showing, here is that there could be millions of bacteria, right, that are in the same kind of colony or environment as this bacteria. So then they would go and they would find kind of an attachment point. Good question. Yes, ma'am. If we're talking about a coronavirus, yes, an M protein would somehow help direct these components that are being put together. All right. So the next phase, right? This is lysogenic. I'm sorry. This is temperate phage, which is lysogenic and lytic. And so here, right, you can see what's interesting is that you have two options. If we follow the blue, that's the lytic cycle that we just talked about. All right, so nothing crazy there. What we also see, though, right, is a lysogenic phase. And a lysogenic phase, this is really interesting because in this case, the viral nucleic acid is incorporated or integrated, right, into the host DNA, right? And this is really cool, right? This is really advantageous because while that viral DNA is hanging out in the host chromosome, it's hitching a free ride, right? Every time that bacteria divides, it makes a copy, right, of that viral DNA because it's embedded within its chromosome. And what's really nice here, right, is that if conditions are good, right, if the bacteria is doing well, it's metabolically very active, there's not a lot of division, or not a lot of threats going on to it, right, this viral DNA can just kind of hang out, right? If bacteria are dividing rapidly, that's a good thing because that means that, hey, it's getting a lot of copies of itself too, right? If cellular division slows down for whatever reason, right, which usually indicates, could be for multiple things going on. Maybe the environment's not as good as it was, who knows, right? But cellular division slows down. When cellular division slows down, all right, that viral DNA, right, can leave the host chromosome, and then you start the lytic cycle that you guys already know, or we already talked about. You guys will know. All right.
So they always end up in lysosomal.
Yes, it's always going to. Yes, we're talking about kind of an exception here. That's a little interesting, but yes, it eventually is going to lead to the cell being lysed. It's kind of a matter of when. Yes, ma'am. Correct. It just chops it up. And sometimes too, and what's really interesting, and in some cases, this is actually kind of random, which is good, right? Because randomness increases your chance of survival. And so sometimes upon initial infection, there's not necessarily even a rhyme or reason as to why it initially goes to lytic or why it goes to lysogenic. But if you have both covered, right, because again, to Blair's point, there are other bacteria here, there are other viruses here. If some are going lytic right away, if some are going lysogenic, then you increase your chances, right, of choosing wisely up front, which ensures that your virus self, right, is going to be able to survive no matter what the conditions are. All right, and again, remember about viruses, we're talking about them being dead, but we're also talking about them being able to respond, right? So it's interesting that something that has characteristics that we would say makes it non-living or dead is able to respond to, right, essentially a change in metabolic activity of a bacteria, right? So kind of weird, but kind of awesome, all right? So a couple important things, right? What's interesting about bacteria and viruses, and I actually wish this was the case in us, is you have something called lysogeny, all right? And so lysogeny is actually protection from another infection. So basically what it's saying is if I was a virus, right, and Jada was a virus and Rachel was a virus, and I infected Morgan, right? What would happen is Jada and Rachel wouldn't be able to also infect Morgan. And again, that's a good kind of, we'll say, evolutionary advantage, because if I've already infected Morgan and Jada, Rachel, and I are all the same type of virus, well, now they, right, can go infect somewhere else. So again, being able to spread out a little bit more. Now, what we've also seen, though, and this is kind of Blair's point about, does it always result in lysogenesis, or does it always result in the lytic phase? And this is where I would say no. So one of the things that we've seen with lysogeny, right, is this idea of lysogenic conversion. And so phage DNA actually has been incorporated, right, and has become a regular part of some bacterial chromosomes, and is actually the reason that those bacteria are pathogenic, all right? So here you have a couple of Clostridium, diphtheria, Clostridium botulinum. So if you've heard of botulism, diphtheria, again, is something that hopefully you've been vaccinated for. Pathogenic E. coli, right, something called the Shiga toxin, which can cause a whole slew of issues. You can look those up if you want, right? S. pyogenes, right? Pyrogenic exotoxins, right? So things that cause fevers. Vibrio cholera, right? The cholera toxin. And so it turns out, right, that we know these are bad bacteria. We know they cause issues. What's interesting is that the issues that they cause are actually from viral infections way, way, way back in the day, right? And I will let you decide how far way, way back is, depending on what view of this world you hold. Yes, ma'am.
So if you know that a virus that you have is a bacteria phase, would taking antibiotics help inhibit the spread of the bacteria?
Okay, say that again.
So if you knew that like a virus that you have is a bacteria phase and was infecting bacteria in the body, would taking an antibiotic help?
Well, I mean, yeah, if you kill the bacteria, definitely. But, you know, I have not looked, Jennifer Buckner, if anybody is, you know, when you go to the doctor, if they're like taking blood and then checking for bacteriophages. You know what I mean? And, you know, one of the things, and I mean, that brings up a good point, we won't spend a lot of time on this, is that we talk about the microbiome a lot. So it turns out you also have a virome, even in your gut. And so there are viruses that are resident in your gut that are interacting with bacteria and interacting with your cells that are also contributing to the state of your gut environment. And we have almost zero idea as to what's going on there. And so presumably some of that is bacteriophage driven, right? But if you think along that line, which is good, right? Well, if you're afraid a bacteriophage is going to make something pathogenic, you kill the bacteria and then you're fine, right? But then again, you're also killing a bunch of the good bacteria. And so it's kind of, and actually before antibiotics, and we won't really talk about it, before we had antibiotics, there was actually a lot of research that was being done on using bacteriophages as a treatment for bacterial infections, right? Because they're a virus that targets bacteria, and they presumably kill most bacteria, although, right, not all of them, which I don't think we had a grasp on at the time. But that still is an area that some people are doing research in. Can we use bacteriophages, right? It's going to be harder for them to develop resistance to a virus than it is to some of our medications. But then you stand the risk of, right, pathogenicity being inferred upon a bacteria, which could be bad. And like I said too, just because a bacteriophage doesn't infect you now, doesn't mean if we started using it as a treatment, right, that, we can see with like the SARS-CoV-2 is a great example, right? This is a zoonotic virus, which means it came from animals, and presumably multiple animals mixed together and then made the jump to us, right? So if those things had never been brought together, we wouldn't have the pandemic. And so you can make the same argument potentially, I guess, with bacteriophages. If you start bringing a bunch of things together purposefully, who knows, right? TBD. Maybe not an issue for us, but maybe an issue for our kids, right? So, you know, again, the botulism toxin, which is from a bacteriophage, used to be a huge issue in canning. Not that big of a deal now because we have a pretty good canning process, but this is one of the reasons, and hopefully you already knew this, but sometimes you have to be a little weary of cans that are dented really bad because there's a chance, right, that something got punctured and maybe something was introduced to that can. Or if you find a can that seems to be expanded, okay, that means that something's living inside and has already been eating your food, so you probably shouldn't eat it, right? But the botulism toxin interferes with the nervous system, and so here you can see, right, this patient has an inability to hold open his eyes, so it's basically causing paralysis. And where you start to have huge issues is when you start to cause paralysis in muscles like your diaphragm, right? That's not good. Those of you that haven't taken A&P, that's the breathing muscle, all right? But what's interesting is that, and you know, not that I'm picking on Demi Moore too much, but you can see here, this is when she's 30, this is when she's 46. And so a lot of Hollywood celebrities use Botox, right, to actually help, right, in directed sites to cause muscle contractions, right? And those of you that have really bad migraines, or if any of you do, and I haven't really looked to see how this works yet, but the botulism toxin is actually being used now to treat migraines, which is pretty interesting. Yeah, I don't know, it's just really cool, things that you think are bad. So there's this bacteria, Toxoplasmosis gondii, I can't remember, it causes mad cat, or infects the brain. So if you heard of like mad cat ladies, when they have a bunch of cats, it's spread via cat feces, and they're actually using the toxin from that bacteria to help treat Parkinson's patients, right? So again, these things, when directed, are used differently. You know, really cool. You can look at this if you want. I'm not going to test you on this, but these are links that are really cool, because remember I told you you can't see viruses under light microscope. Turns out that's a little bit of a lie. As the permafrost layer up north has been thawing, we have been discovering new interesting things, and one of the things that we've discovered are actually giant viruses that have been trapped in the permafrost for a long time, and they're actually big enough to see with a light microscope. The thing too, if you watch this, is if any of you are into horror movies or zombie things, this kind of made me cringe, because they've actually been reviving these giant viruses, and have been, but they've assured people that they only affect amoebas, right? Which I feel like is how every zombie movie starts, right? Oh, it only infects this, right? Okay, so, but it's really interesting, and I'm not saying that global warming is a good thing by any means, but it has actually revealed some things that we never would have discovered, you know, before. Again, if you adhere to global warming. Also, not having that debate. All right, we can have a global warming evolution virus debate sometime in my office, right, during office hours. All right. So the last phage that we're going to talk about here before we move on is the filamentous phage. So this actually requires something called an F. pilus. We haven't really talked about that yet, but an F. pilus, you'll see here in just a second, is an important component on bacteria that can undergo something called conjugation, which is a fancy word for bacteria sex, right? This is actually one of the ways that bacteria can share genetic information. So this is the F. pilus. Presumably, this F. pilus would be used to make contact with another bacteria, all right? And when that F. pilus makes contact with the other bacteria, right, this bacteria would be able to pass along either a plasmid, right, that small segment of DNA, or in some cases actually a whole chromosome, right? So again, it's not true sex, right? If we talk about, you know, life being created from humans having sex, right, when you get half from mom, half from dad, boom, baby, right? But right, this is kind of close for bacteria. And so what's really interesting is that a bacteriophage has learned how to take advantage of this. So the F. pilus is actually, right, what it attaches to for entry. And then you'll see down here, it is actually what it leaves through also, all right? And so I don't know if this helps Alanis or makes her more confused, but this is a really good example of kind of budding here. So you see, right, these capsomeres, so those are individual components of the caspid, have been sent to this part of the cell membrane near the F pilus, right? So they're kind of waiting there. And so basically what they're going to do is they're going to, it's basically going to bud off through the F pilus, and it's going to take these proteins that it's put in there with it.
So will
Okay.
that be host DNA too with that?
No, that'll only be, in this case, this would only be viral DNA. And we will talk about next unit. I haven't heard of it happening in filamentous phage infection, but in alytic and lysogenic, it's very possible that host DNA can be packaged into a virus. And we'll talk about why that's important next unit. So what's something that you can point out really quickly that looks different from this infection compared to a lytic or a temperate? Okay, so yeah, what you're seeing here, yeah, so the DNA is staying in its circular form, right? Okay, so that's true. That's good. What else? The bacteria is not dying, right? So this is something that we call a productive infection. And it's productive, right? Because you're not killing your source of replication, all right? Now, what happens is that this form of replication is much slower, all right? Because basically you're allowing the bacteria to do its regular thing while you are also trying to do your regular thing, all right? So it would be, you know, I guess kind of if you had a bunch of people over, you know, for a party and you were planning on doing dishes that night on your own, you had an unexpected, we'll say a presidential debate party, and then you're still one person, but you're doing twice as many dishes, right? So you're gonna go a little slower. And so what this does is it slows up replication of the virus. It also means that the bacteria is dividing a lot more slowly, all right? Because you're basically using one set of machinery for two different jobs now. But, right, it could be good because if you don't kill your host, then you can keep on making copies of yourself. Okay. Okay. And actually don't worry about horizontal gene transfer here because we're going to talk about it in the next unit. All right, so I'm going to save that for the next unit, right? But that's what I was talking about with Blair. Sometimes host bacterial DNA can actually get packaged in this virus, all right? And we'll talk about what that does next unit. So, you know, one of the things that I want you to appreciate, there we go, is that bacteria defend themselves, right? And so we spend a lot of time, I think, talking about our immune systems, right? But what we'll see here in just a second is that bacteria, right, have their own ways of defending themselves. So one of the most fundamental things that they can do is they can block the receptors on their own surfaces. Those of you that took or have taken ANP1 or remember anything about enzymatic activity from previous classes, remember we have something called competitive inhibition and non-competitive inhibition. If you remember in competitive inhibition, right, you are blocking the active site of an enzyme, right? You're filling it up. So whatever that enzyme wants to interact with, it can't. Same kind of idea here, right? You block your receptors on your surface, and that means the virus can't interact with those receptors that it might use to attach itself, right? So Staph aureus, which you guys have used in lab, I think on a regular basis. In fact, I was in one of your lab groups yesterday, and was it your group? Yeah, when Professor Perry asked about, I think, MSA plates, and the silence was even awkward for me, right? And that's saying something, right? But then someone eventually got it, right? But S. aureus produces something called protein A, all right? So they produce this protein, it's released, and then it actually blocks receptors on the surface of that bacteria, which prevents bacteriophage attachment, right? One of the things that we'll talk about, but I know even Professor Perry was talking about yesterday in lab with Klebsiella pneumonia, right? It's kind of mucousy. It's got a good capsule on it, right? And so a capsule is basically a sticky, snotty coat that's around a bacteria, and that actually can trap the viruses, right? And not allow them to attach to receptors on your surface. Kind of the same basic principle, right? That the mucus, right? Your GI tract, respiratory tract is playing. It prevents attachment, all right? Viruses, bacteria, they get stuck in that mucus layer in you, and they can't interact with your epithelial. Same general principle here, all right? Those of you that have had genetics, right? You used restriction enzymes to pop that segment of, what is it, jellyfish DNA? Is that what you guys use, right? Into that E. coli plasmid. And restriction enzymes are really cool because what we've found through research is we know kind of exactly where they like to cut, right? And so if you have the sequence of a plasmid, right, you can look at that plasmid and you can say, okay, if I add this restriction enzyme with it, this is where it's going to cut. And so bacteria produce these restriction enzymes that cut up viral DNA that enters. Why is that potentially an issue, though? And you can, I mean, even look down here and take a guess. Why is that an issue? Well, if you cut it? No, because you're going to interrupt that. Don't think about the virus, think about the host. Could cut its own, right? I mean, it'd be like if you gave a kid that was 7, that's hopefully smart enough to not run with scissors. You gave him scissors and you put him in a room full of important papers and you left him there. Shame on you, right? He's going to cut everything, or she, right? I can speak from experience now with a daughter. Girls can be just as, you know, well, you all know, right? And so it's kind of the same thing here. Like these restriction enzymes, they're just cutting DNA. And so what we see here, though, is that some of you have heard of methylation, or maybe you've talked about that before, right? It's basically targeting DNA with methyl groups. And what we see, and this is even the case in us, the older your DNA is, right, the more time you've had to add methyl groups to it. And so viruses, right, when they infect, that's fresh DNA. It hasn't had a chance to be methylated. All right, so these restriction enzymes actually leave methylated DNA alone, and they target, right, non-methylated DNA. All right, so they're going after something that's been introduced. The last bacteria defense that we'll talk about is something called CRISPR, which you guys have probably heard of. CRISPR is actually being used in an attempt for genetic modification, potentially future in gene therapy. But its original purpose was actually basically kind of I'd say a little bit of a adaptive immune system equivalent in bacteria. And so what you see here, right, is you have a phage infection. It searches DNA. You then have something called a Cas protein. This Cas protein chops up the phage DNA, right? It's able to distinguish and say, hey, this is phage DNA. It chops it up. And then, right, you have this CRISPR array within the bacterial chromosome that has these small, basically little openings where you can insert small segments of viral DNA. And so basically what you see here is each color is indicating a different phage infection, right? So this bacteria has gone through a lot. But then what we have happen here, right, is this CRISPR array, right, gets transcribed, right? And so you basically make these little CRISPR RNA components that are related to previous infections. They get Cas protein complex, and then you're able to kind of move around and scan and say, is this viral, right? Or is this host, right? So it's basically like a little identification, right, for viral infections, right? So it's kind of cool because it actually indicates, right, a past infection history, right? So it's kind of a very primitive form of immunologic memory.
Can you say what you said after it chops up the phage DNA?
Yeah, so it chops up the phage DNA, and then a small segment of that phage DNA is added to the CRISPR array, right? So it's basically keeping a history of previous infections or an ability to identify, well, keeping a history of previous infections so it can identify future infections, right? Okay, we don't have much time here. I'm going to go through this. Let's see how many slides I have left, because I want you guys to know this. Okay, there's not too many slides left. So whatever I don't finish here, I want you guys to look at, but I promise it's not overwhelming. It's just a slight shift, right? And we're going to talk about these 5 steps here in just a second. So we've talked about bacteriophages. We have mentioned the coronavirus, but we didn't really talk a ton about it. And so what you'll see is there are a lot of similarities, but some slight differences between the infection cycle of an animal virus versus the infection cycle of a bacteriophage, right? So attachment, genomic entry, synthesis, assembly, and release. So in principle, right, those 5 steps are the exact same. How they happen is a little different between the 2 kind of different types of viruses, right? So here we have attachment and genomic entry. And what you can see here, right, is that this is showing 2 enveloped viruses, but even here, entry by membrane fusion could also occur if you're a non-enveloped virus. So that's one of the first things I want to point out, though, regardless if you're enveloped or non-enveloped and you're an animal virus, is that you are gaining entry into that cell. Remember, the bacteriophage, right, it's injecting its nucleic acid. In this case, you're bringing in the whole viral particle. Here you have, right, your receptors. Here you have spikes, just 2 different ways, right? So membrane fusion, you just kind of pop your way in. Here you have endocytosis, right? So you basically trick the cell into eating you and engulfing you. No matter how you gain entry, though, right, that caspid's going to break down. You release your nucleic acid. That nucleic acid is then put into the cellular machinery, right? With RNA polymerase, maybe DNA polymerase, right? You have your ribosomes. All that stuff starts to come into play to make copies. Okay. Again, don't get overwhelmed here. This is the 3rd step, synthesis. One of the important things that I want you to remember, right, is that you're always trying to make mRNA so that you can make protein. I just like this chart because it's saying, hey, here's what you start off with, DNA or RNA. Here's a interesting group of reverse transcribing viruses, which is actually what the HIV virus is, right? No matter what you're starting with, right, you're always trying to make protein, right? And so this is actually what the coronavirus is, right? They're single-stranded positive. So this is what the coronavirus is right here, right? So the thing that's cool about the coronavirus, as soon as it enters, right, it can start making protein. So again, we've already kind of talked about this a little bit. Nothing too crazy here. All right. So what's really cool about the assembly, right, is that it appears to be somewhat spontaneous. So we do have that M protein that helps direct it, but we're still not quite sure what triggers the assembly and how viruses know what parts go where, right? Because for the most part, they have features that that we associate with them being dead, right? So there's a lot of unknown things that are occurring. Now, what's really cool too is that, again, components are things that make them alive, right? Viral genome can be altered as we're actually assembling that virus, right? So there's a chance for change and there's a chance for mutation, which is good for them and bad for us. All right, again, release. So Lannis, maybe this will really help you with budding, all right? From an enveloped virus. Now, if you're not an enveloped virus, you're primarily going to be released through a lytic, right, phase, right? So you're going to build it up, you're going to burst, and it's going to release you. But here you can see you have viral proteins that get embedded within the cytoplasmic membrane of the host. The virus is attracted to them, right? You bud off. And so now part of that envelope is actually part of the cell membrane that has viral proteins embedded within it. Okay, this is really, really cool because it doesn't trigger the immune system as rapidly, all right? Because that cell will eventually naturally die from being overworked. but it won't explode. When things explode, whether they're cells, right, or other things, they attract attention, all right? And so if you can cause something to not explode, it doesn't attract the attention of the immune system as well, and then that virus is able to kind of sneak past, all right? What I want you guys, this will be where we stop. I won't do prions on this exam, all right? I'll stop here with viruses. Just take a quick look at this. It makes sense. Acute infections, chronic infections, latent infections, right? So SARS-CoV-2, right, is an acute infection. We think, right? If you talk about cases of, yeah, right now we think it is, right? You get it, you get better, and then you're done. Doesn't mean that you don't have complications afterwards, which you guys have heard about, but you can kind of see these different categories of infections here. So this will be where we end for your exam, right? Don't worry about prions. All right, get out of here, clean your space. Have a great